Hi, I'm Sicheng Li. I'm glad to show you our new fNIRS finding of speech. This study is about the cortical hemodynamic response and functional connectivity. So let's begin. Now the video shows a state-of-the-art Speech BCI that decodes brain activities to speech sound. Our research is the first phase investigation for non-invasive active Speech BCI. It has a broad perspective. fNIRS is a non-invasive modality. It has several advantages, such as relatively higher spatial resolutions, portable, and so on. Previous fNIRS studies used paradigms involved in inner speech processing. However, there are lack of explorations of the cortical response and functional connectivity in speech imagery without external cues. Besides, whether and how the dorsal sensorimotor cortex and Broca's area are involved in speech imagery remains unclear. So, our study has two goals. First, to validate the fNIRS reliability. We used our speech as a control group, and we compared with other studies. Second, to characterize the speech imagery pattern for fNIRS speech BCI. We mapped the cortical response, evaluated brain regions, and captured functional connectivity. This is the paradigm. The paradigm consists of relaxed preparation, 20-hour trials and 20 cover trials. Each trial has a 2-second prompt, 15-second execution, and about 13-second relax. We recruited 13 subjects for experiment. We divided our coverage into 6 regions: dorsal and ventral sensorimotor cortex, secondary motor cortex, Broca's area, and dorsolateral prefrontal cortex. We convert our fNIRS light intensity into task-related hemoglobin concentration changes. We used bandpass filtering and functional component separation to denoise. We calculated cortical activation and functional connectivity. As a pre-processing result, the ground averaged response is consistent with the expected one. Here is the typical channels time series. Significant activations were shown in motor cortex and in Broca's area. As a preliminary finding, the overt and covered speech show different cortical response patterns. Here is the overt speech Hbo mapping. The bilateral VSMC, VM2, and Broca area was most significantly activated during the overt speech, and the bilateral DM2 was less significantly activated. The cover speech mapping, DSMC and DM2 were significantly activated, whereas Broca was deactivated during the cover speech. This might be a surprising finding, as since DSMC is usually involved in hand and arm movement, not the articulator movement. But some study verified DSMC is also involved in imagined vocalization. For the region-wise Hbo response comparison, DSMC had a significant regional differences and large engagement range, whereas DM2 did not. For the channel-wise comparison, the dorsal motor cortex preferred to cover speech, whereas the ventral motor cortex and Broca's area preferred to over speech. At last, DSMC-related functional connections were enhanced during cover speech compared to over speech. Here is the summary of cover speech, over speech, and functional connections. These findings are insightful to state detection. We further suggest about the channel layout and decoding features of Speech BCI. At last, thanks to my mentors, group members, the institutions, and the findings. Okay, thanks for listening.